All right, everybody, it is 12 o'clock, so we're going to go ahead and get started. I'm Professor Heckman. Welcome to biology. You will not be seeing my face today because I am currently recovering from a severe allergic reaction to the medication that was given me to treat my first illness, and my face doesn't exactly look like a face. So you'll get to see me another day. I also included a little picture on the PowerPoint. So we're going to start the semester with this quote: "Science, my lad, is made up of mistakes, but they are mistakes which it is useful to make because they lead little by little to the truth." And I think this is a beautiful representation of science because a lot of the time, especially when people do experiments, they do what they think will get them the quote unquote right result. And if it doesn't turn out that way, they think their experiment has failed. But in reality, you can learn a lot more from a failed experiment than from a successful one, because you can look at your data, you can look at your experiment and say, where did I go wrong and why did this happen? And that's what science is all about. First of all, if you're sick, stay home. We don't need any sick people in the classroom. We do track attendance, but that is only because NWAC requires it as a school. But do remember to come when you're healthy, of course. Regular attendance is essential for a passing grade in the course. This is me. This is what I looked like in the summer. Well, I guess I still look like that. And this is my special interest. I love turtles. I have a turtle rehabilitation center that I'll be telling you guys about in a little bit. And this is a giant Galapagos tortoise at the Oklahoma City Zoo. And I was so excited to meet it, I almost cried. This is my family. That's my dad. He's Dr. Heckman, and my mom is Mrs. Heckman. Don't call me Mrs. Heckman, please. It makes me feel old. That's my brother. That's Matthew. He's Mr. Heckman. Then we have Luke and Rebecca and Biz and Esther, and then that's me, and I am Professor Heckman. And these are the two most important men in my life. On the left is Wolverine, and on the right is Loki. They are little terrors, but they're cute, so they get to stay in my house. Throughout the semester, you will notice that I sweat a lot. I sweat when I'm sitting, I sweat when I'm cold, and it's a condition called hyperhidrosis. And you can't see them. No, what do you see? I see nothing. That's how I feel in my life sometimes. Okay. Oh, share. There we go. Is that better?
Yes.
Awesome. Okay. So let's just go back briefly. That's me, my family. These are my cats. And this is, how do we admit people? View lobby. Admit. Cool. Okay. It's called hyperhidrosis, and I know that a lot of you people are in this class because you want to get into the nursing program. 3% of the US has hyperhidrosis, so if you are a nurse, it is probable that at some point in your career you will see somebody with hyperhidrosis. So I thought I would take the opportunity to tell you a little bit about it. Basically, it's excessive sweating that occurs independently of temperature or exercise. It can happen at any time, any place, for no reason whatsoever. Risk factors include genetics, if you had repeated febrile infections as a child, a history of tuberculosis, or if you drink a lot. Those may increase your risk of hyperhidrosis. There are a lot of celebrities with hyperhidrosis. We have Charlize Theron and David Hasselhoff and my brain's drawing a blank. Halle Berry right there on the Ellen Show. We've got Steve Carell and Robert Pattinson and Chrissy Teigen. And they've all spoken openly about their hyperhidrosis and their struggles with it. Let's talk about the two kinds. There's primary and secondary. Primary is a malfunction of the nervous system. We've got eccrine sweat glands and apocrine sweat glands in our skin tissue. And where's my mouse? I need a pointer. There we go. All right. And the eccrine sweat glands, those are ones that are not attached to hair follicles. And when they're triggered, they just put sweat directly onto the scalp or the skin or, you know, wherever the pore is located. And nerve signals that are faulty triggering that will be part of hyperhidrosis. Secondary hyperhidrosis is a side effect of something, a medication or another condition. And I have primary hyperhidrosis. Unfortunately, there's no cure, but you can change your lifestyle to accommodate your hyperhidrosis using powerful antiperspirants or eliminating everything good in your life, including spicy foods, caffeine, and alcohol. These people are having a great time. My cousin has this as well. It's genetic in our family, and this is what she does. Her treatment, she puts her hands in these little pans of electric lights and runs electricity through them. So she's basically electrocuting her hands. It works for her, so good for her. But yeah, that's one way you can treat it. You could also get Botox injected in your underarms. That's another temporary treatment. It numbs the nerves that go to the sweat glands. Or you could get your sweat glands removed. There's surgery. You have laser surgery and then more invasive surgery that can go in there and actually take out the sweat glands so that will remove your hyperhidrosis. Or you could use microwave therapy to zap them. And that's my bibliography. Whenever we do presentations in this class, you're going to actually, your final presentation is going to be your final project, and we're going to cite everything in APA format, but we'll get to that later on. So don't worry about APA format just yet. All right, so how to succeed in this class. Come to class and pay attention or take notes. You will need a laptop. If you do not have a laptop, there are some in the classroom available for classroom use only. You can work on those and then save your items to a flash drive or something and take that home with you. But you will need a laptop because what we do, except for today, I put student versions of this PowerPoint online. You will download that student version before class. You will follow along, and there's going to be blanks in there, and you'll fill in those blanks from the main PowerPoint that I will be going through. And then you will use that as your study notes for the tests. It also helps if you read over your notes after class, just briefly. Science says that you need to see something at least 3 things in order to remember it. That varies obviously from individual to individual. I have to see something at least 10 times in order to remember it, unless it's a turtle fact, then I remember it the first time. And number 3, use study guides to prepare for the test. Don't start the day before the test because there's a lot of stuff on the tests. I have already put up the study guide for test 1, which is on Canvas. There is a module called study guides, and it's divided into chapters. So after today, you can go download that and fill out everything from the chapter 1 section, which I think is pretty short. Let's see. Yes. Do a few items every day. Syllabus. The syllabus, it is long because it is my contract to you. It includes exam makeup procedures, requirements for assignments, participation and attendance policies, all the things that we are both going to abide by while we're having this class. You are responsible for knowing all that information. If you want to record class while we're in-person class, meet with me and we can work something out. And let's go over some highlights of the syllabus right now, shall we? Why is there a little 1 down here? What does that mean? Okay, well, I'm not very good at computers, so here's our syllabus. You guys are section 14. Don't be afraid to ask for help when you need it. This is my email address right here. My last name has 2 N's on the end, so be careful to put 2 N's instead of just 1 N when you write Heckman. Otherwise, it'll go to someone else. But yeah, I became a teacher to watch my students succeed, not to watch you guys fail. So if you ever need help, don't be afraid to ask. You're going to need a computer or a laptop. We already discussed that. Microsoft Word and PowerPoint, you need these so that you can edit the PowerPoint presentations while we are having class. And this gives some instructions for how to download them. The lab manual, we have all talked about, we've talked about the lab manual already. The issue has been the weather. I know that some people have tried to print it using the grab and go option, but you haven't been able to pick it up yet. And so I changed the due date. I want you to bring your printed lab manual on the 29th, okay, which is the day that we are going to begin our next, yeah, 29th. That's a week from today, the day we're going to be doing our second lab. And you can get credit for it at that point. But for today, I have put an interactive version of the handout online, so you can type in that, or if you have it printed out, you can write your answers on there and use the graph paper there. That will be helpful because you'll have to make a graph and then take pictures and upload those. But yeah, you're going to have to bring your lab manual for the labs. Printing, nothing will be provided for take-home use in class, as I cannot afford to print everything for everybody. So here are some ways that you could print things for yourself. COVID formats, if there's somebody that has COVID, obviously don't come to class. Here's some goals, learning outcomes, exams and quizzes. They will all be taken in person on the scheduled date, with one exception, which we will discuss in a minute. And we'll go over this exams and quizzes in more detail. So we're going to come back to that. Labs. Each lab requires setup and use of specific materials, some of which may be dangerous or perishable. They are provided by the school only during the lab sections, and because of this, makeup labs are not possible. However, I understand that things come up and you may need to miss a lab day. Because of this, I dropped the lowest lab score. So if you get sick or your child gets sick or something like that and you're not able to come to lab, if you get a 0, that's going to be your only 0, and it will not affect your score because I dropped the lowest lab score. And I dropped the lowest exam grade, not the final though. When you turn something in, please write legibly so I can grade it. If you miss a class, contact a classmate to find out what you missed. We're going to take care of this when we can get to an in-person class. There's a little icebreaker I like to have you guys do in teams, and then you exchange phone numbers so you can form study groups, and that's really helpful to have other people's information. Okay, cool. We do sometimes do things in class for a grade. If you are not there, it is your job to find out that something has happened and to ask if you can complete it. Some things can only be completed in class because they're group work, but some things you will be able to make up. So make sure that you find out if those things happen. Sleeping. If you are tired, you can sleep, but don't sleep in the classroom. This is because sleeping in a classroom is ineffective. It's really hard. Nobody sleeps well sitting up. The lights are bright. You've got that overhead light buzzing noise. Go find somewhere soft, lie down, take an effective nap, and come back when you feel ready. Sleeping in the classroom just does not do anything good for anybody. Be respectful, act like an adult. Participate by paying attention, taking notes, contributing to class discussions, completing the assignments, and taking part in the learning activities. Come to class on time. This is a big one because sometimes I will give quizzes at the very beginning of class, and I will also give announcements at the beginning of class. And if you come to class late, you might miss those things. So then again, it's a good idea to have the information for some of your classmates, the contact information, so that you can, like I know people form group snaps and things like that. Plagiarism. Plagiarism will not be tolerated, especially on your final project, which will be a presentation about an endangered species. And if you need more information about plagiarism, there's some here. How to succeed in class, we've already done that. Blah, blah, blah. Fully participate in at least 2 classes in person, well, if we can, and complete the lab safety quiz before 1:30, or you may be dropped from the course for non-participation. That would just allow you an 80% refund on your tuition. We have resources at NWAC for students. If you need mental health counseling, that's offered for free. You can click on this link and go to that. And we also have a food pantry on the Bentonville campus. You can find more about that at Student Services as well. And then the very last page of the syllabus is the most important page, which is the schedule. It is your responsibility to know these dates and be prepared for the test. And you have to be dressed in appropriate lab attire on lab day. Your section 14, your final will be on the 6th of May from 12:30 to 2:30 p.m. So that's our syllabus. Does anybody have questions so far? No? Okay, cool. All right, let's talk about exams and quizzes. We're going into a lot of detail about this because they are very important for your grade. And I just want to make sure that my expectations are clear and that you guys know what you are expected to do and how you can succeed. So we will take exams on computers, but you have to be in the classroom. That's an NWAC policy because it's an in-person face-to-face class. You have to be in the classroom taking the test. We will use the HonorLock proctoring system. Most of you are familiar with Lockdown Browser. HonorLock is similar to that. You can bring your computer, or again, you can use one of the laptops in the classroom if you need to. If you do not take an exam, you will receive a grade of 0. And you can miss an exam per semester and make it up, but you can do that for only one exam. And in order to make it up, you must contact me at least 48 hours prior to the exam to schedule an alternate time when you can take the exam. However, if you have an emergency the day of the exam, you must contact me by email before the start of class. This means the timestamp on your email must be before the start of class time. If you contact me any time later than the start of class, then you cannot make up the test. And you can, for sure, use your phone to email me. All right, we're going to do some clicker questions in your Google Play Store or Apple, whatever the Apple version of that is. Go to Socrative Student and download that app. This allows your phone to function as a clicker device. And this is how I keep up with not attendance, but participation in the classroom. It helps students to stay engaged, and it also gives you an opportunity to use your phones because I know that we all love to use our phones. And it's going to ask you for a room name. Your room name is going to be NWAC Bio. Yes. Now I have to log into mine. There we go. Yes, okay, awesome. So hopefully, oh, I hate this thing. Okay. Here are some clicker questions. Hopefully you have that downloaded. If you don't yet, we're going to have quite a few clicker questions, so join us when you can. First clicker question. A student approaches me the day after the test and says, my mom had to go to the hospital yesterday and I couldn't contact you. When can I make up the test? Will this student be able to make up the test? Go ahead and answer on your device according to the answer choices. All of these things are real scenarios that have actually happened. Oh, interesting. Okay. 38% say yes, and 62% say no. The correct answer here is no, but there is something he could have done to make himself eligible to take the test. What is it? Excellent. 100% chose B, which is the correct answer. Yes. Obviously, if you have to take your mother to the hospital, you have to take your mother to the hospital. But you are also a student, you're paying money for your education, and you can invest a few minutes to send a quick email. What about this student? A student does not take the test, no contact with the teacher is made. Will the student be able to make up the test, yes or no?
What was the name of the app?
Socrative. Socrative student.
Okay. Thank you.
Uh-huh. Yes, 100% chose B, which is the correct answer. Very good. And here's our last one for a bit. Yes, 100% chose A. Good job. You guys are on top of this. If I have COVID-19, well, obviously I'm going to stay home. Something that I do to help you guys keep up with the material and review is sometimes I will give you short quizzes before the class period, or well, before the class material starts. There'll be five questions, very simple, covering the material that we covered the day before, and I give you the answers to these questions. The reason I do this is to help you review and help you get an idea of what's going to be on the test when the test arrives. So in order to get those answers, you have to be in class on time the day that I'm giving the answers. If you miss a quiz, you are able to make it up, but you have to be sure to ask me for it. And if you miss a class, you are responsible for finding out if there is a quiz the next class. I am not responsible to track down and tell you. So make sure you keep up with that. And all you have to do is memorize the answers and then write them down the next day. You're responsible for printing all class handouts. We talked about that. The PowerPoints on Canvas, we talked about that. I know that some of you have come in since then, but basically I provide student versions of the PowerPoints. And this one today, I did not because I recognize that most people only have one computer screen and you can't like type on one thing and watch one thing at the same time. Well, maybe you guys can, I can't do that. But there will be blanks. And so you'll fill in the blanks in the student version of the PowerPoint based on the teacher version that I am presenting. And then those are the notes that you will use to study for the test. If you want to read the textbook, it has a lot of good material in it. It's where I take my information from when I make my lectures. I am not going to require any reading for this class because I understand that some people cannot afford the textbook. But if you can afford it, if you want to get it and you benefit from reading, I have created some active reading guides that are going to be on Canvas for each chapter. You can complete those. Those are optional just for you to do to help sort of guide your reading as you are reading. Lab safety. Yes, let's do the lab safety PowerPoint. I know that most of you have, where's my paper? Yes, most of you have done the lab safety quiz, which means that you've gone through the PowerPoint, but I'm still required to do this by the department. So let's go ahead and do that. Where is that going to be? Right here. Principles of Biology Safety Lecture. Yay! The ground rules. No food or drink is allowed in the classroom because the classroom is used for chemicals and you never know what somebody may have left on the table before you. On lab day, you must be in pants and closed-toed shoes. We have some scrubs and some booties that you can put on over your clothing if you forgot to dress appropriately. But if we don't have enough for everyone, then you will have to go home and change. This is just a safety issue. If you're in doubt about a proper laboratory procedure or given task, you should ask your instructor before proceeding. Lab stations. This is going to be your desk. We're going to use the desks as our lab stations. So keep them free of extraneous items so that you don't get things cluttered. And after your lab is finished, you should clean your lab station with, well, we don't use Simple Green anymore. We use a hospital-grade disinfectant, which eliminates the COVID virus. So wherever it says Simple Green, we're going to use disinfectant. Lab equipment. Always make sure that at the end of the lab, everything that you used is rinsed, wiped down, put back where it's supposed to be. Lab machines should be turned off. Test tubes should be rinsed and placed in the test tube bucket. It's very important. I will put a cleanup slide for each lab on the board, and it's very important to follow that, especially when we're disposing of different chemicals. It will tell you how to do that. Shoes with cutouts or vents that leave the skin exposed and unprotected are not acceptable. You cannot wear pants that have tears in them or holes. You're going to have to pull on some scrub bottoms over your pants if you wear those. Also, if you're wearing anything that is really tight, that's not recommended as well, because if you spill something on yourself, the fabric is going to suck it up. And if you're wearing like a legging or something that's really close to your skin, the fabric will then hold that chemical close to your skin. Basically, don't wear loose-fitted clothing because your clothes could drag through things and knock things over. And if you have any jewelry that you like, don't wear that. Don't wear any dangly jewelry. Basically, don't wear things you are not willing to part with, because if you get a hazardous chemical on your clothing, we will have to take it and send it off to be properly destroyed. Also, please do not wear high heels or wedges. It gives you a risk of tripping and falling over onto somebody's lab station and smashing glass, and then you're going to be covered in chemicals, and that's not cool. If you have long hair, tie it back when you're in your lab, obviously. PPE, this stands for personal protective equipment. That is goggles or glasses. If you have glasses already that you wear, you can wear those and you don't need to put on glasses over those glasses. But if you don't wear glasses, you will have to wear the lab glasses provided by the lab. And gloves, always wear gloves. They are provided free to you at no cost. You can use as many pairs as you like. Remember, the gloves are to protect you from any hazardous chemicals you're working with that could burn you, poison you, etc. Yes, we use nitrile gloves. If you need a different kind of glove, let me know. Nitrile is completely latex free, but we do have other kinds of gloves if you require other gloves. Remember, while you are wearing gloves, if you touch anything and then you go to touch your phone, whatever you just touched is now also on your phone. So you need to be cognizant of that during lab time. Another thing is that sometimes we use timers. Timers will be provided for you. You are able to use the timer on your phone, but the college is not responsible for anything that may happen to any of your belongings during a lab, which is why it's really important to be very careful about those things. Yes, gloves, nitrile is what we use. Goggles versus safety glasses. I think safety glasses are a lot more comfortable, and that's what we have. So there we go. Do not take off your PPE until you are done with your experiment. A lot of the time, I see people get done with their experiment, take off their gloves and their glasses, and then use their bare hands to clean up their dirty glassware. And that just completely defeats the entire purpose. Like, you might as well not have been wearing gloves at all the whole time. So make sure that you wear your gloves until everything is completely cleaned up. Wafting, if you need to smell a chemical, don't stick your nose over it like this guy is doing. Instead, use your hand to gently draw some vapors towards you, and that'll allow you to smell the chemical safely. This guy says, I don't always smell chemicals, but when I do, I waft them. Chemical reagents. A reagent is basically a chemical that you're using for a chemical reaction. They should be transported with 2 hands. Don't gather up a huge pile in your arms and stumble around the classroom and then, plop them all down on your lab desk, because you could drop some of them, and we don't need toxic chemicals on the floor. Never carry more than 1 chemical at a time. If it's not labeled, don't pick it up. If you spill it, let me know. SDS sheets. These are called safety data sheets, and it tells you the dangers associated with each chemical. And there will be a binder of SDS sheets on the lab cart for the chemicals that we are using for that particular lab. If you are interested, you can read those. And then we also have a full binder in the back of the classroom for all the chemicals we use throughout the class. Broken glass. We have a broken glassware bin, which I will show you where it is the next time we meet in person. Do not pick up broken glass with your hands. There is a dustpan and little sweeper, and use that because you don't want to cut yourself. If you have cut yourself, oh wait, okay, if you contaminated it with blood by cutting yourself, all of those things need to go in this red biohazard container, which is also in the classroom. Any injury, no matter how minor you think it is, should be reported, because even tiny little things could become a big deal later on, and we just want to have documentation of everything that happened. So if you have an accident, let me know, and we will fill out a report and just submit it to the lab supervisor. You'll have to sign it and stuff. If you are cut by broken glass, have someone else get the first aid kit for you. You don't want to go get the first aid kit, because if you do that, then you're going to get your blood on the first aid kit. Have a friend go get it, and then that friend can tape you up with a Band-Aid and let me know, obviously, so that we can do an accident report. If you spill a chemical on exposed skin, you should flush the exposed skin under the cold water for 2 minutes. Let me know, and then we will reassess after 2 minutes. Eye wash station. We have this in the back of the classroom. You will basically flip up these little yellow covers that are covering the spigots. You push the lever towards the wall. Water will start flowing out of the spigots into your eyes, and you hold yourself there for at least 2 minutes, and then we will reassess after 2 minutes. The safety shower is like the last resort. You do not want to have to use the safety shower because there is no shower curtain. But if you have to save your life, you're going to use it. And I'll show you how the next time we are in the classroom. Oh, wait, here's a picture. Awesome. So it looks like this. There's the eye wash station. You go stand under it. Everybody leaves the classroom except you and a friend. You strip, you take off all of the clothes that have been contaminated by the chemical, and then you pull this triangle downwards, and that will start the flow of water, and you stand there for 15 to 20 minutes. There is a fire blanket in the classroom that can be held up as an impromptu shower curtain, and that is something good to keep in mind if you end up having to use the safety shower. If there's a medical emergency, dial 911. If you are pregnant or immunocompromised, please let me know, because there might be some modifications that we will have to make to the different laboratory procedures. If you are allergic to anything, let me know, because often we use food items in our laboratory procedures. Never pick up a chemical of something unknown. Let me know if you find an unmarked bottle. The fume hood is in the back. This is what the fume hood looks like. If something is in the fume hood, it should stay there. This is just like a self-circulating environment in here that keeps any vapors inside the hood. So if we put like some ethanol in there and the ethanol starts to vaporize, it won't waft out into the classroom. It So what you need to do is if you're going to use chemicals that are in the hood, you need to take your test tubes over to the hood, get the chemicals, and then go back to your testing station. Do not remove things from the hood if they are already there. Use the fire extinguisher on a small fire, which is 3 feet by 3 feet. Obviously, do not use it on a person. We have a fire blanket for that. Stop, drop, and roll if you get put on fire with the fire blanket around you. There we go. There's our fire blanket. To use the fire extinguisher, you use the acronym PASS. You pull the pin, aim at the base of the fire, squeeze the trigger, and sweep back and forth. Again, don't use it on a person. If there's a large fire, get out. Pull the fire alarm, call 911, and we will congregate near the classroom. There's a set of dumpsters outside the building near our classroom where we will congregate, and I have to count everybody. Fire alarm, yep, so we'll just go directly out of the door. Don't clog up the parking lot because the emergency vehicles need to be able to get there, and we have to stay outside until a police officer or school official tells us that they can return to the building. These maps are for the other building, the campus in Bentonville, but if there is a severe weather threat, you could go to the bathrooms or to the area where the vending machines are. Those are both considered safe spaces. Sorry, that's my cat. I try to encourage them to, you know, play with their toys instead of my stuff. So that's what he's doing. He can continue to do it. All right, active shooter. If there's an active shooter, run, hide, or fight in that order. If you can get out safely, leave. If you cannot, remain quiet, lock your door, barricade the entrance, turn off the lights and call 911, and fight, prepare to defend yourself as a last resort. Improvise weapons with the intent of distracting and disrupting the shooter. There is an app called BC Alert where you can get all the major emergency alerts, the weather alerts, the school closings. You go to bcalert.com to sign up, and then it will give you an option of different counties and schools, and you can select NWAC from that list. Before you leave the classroom, the last things you should do are wipe your lab stations down with disinfectant and wash your hands. Are there any questions about the safety PowerPoint? Okay, so let's talk about this lab manual again. So I've reset the assignment to 129, 15 points for bringing in your lab manual. This is the text of the assignment that you have, or the announcement that I put on Canvas. So you have this text. This is telling you how to print things. And we are not going to do every single lab. We're going to skip 2 of them, which is why I told you which pages you need to print. And the lab safety quiz is no longer due tomorrow at midnight. Most of you have passed that. If you did not take it, if you are Ashley Areza, Natalia Cully, Grayler Massey, Jasley Meadows, or Alexa Rigsby, you guys either did not take it yet or took it and didn't score 100%. You have to take it as many times as you need to score 100%. So you will do that today before you start the lab procedure that we're going to be doing today. All right, take a picture of this or something. On 129, bring your entire printed lab book with you. This is worth 15 points. You must show it to me at the beginning of class. If you forget and have to run off to print it, you do not get credit, but you will still have to print it. So you may as well have it at the beginning of class. This is to prevent the printer from getting clogged up because there's a limited number of printers on the Washington County campus. And if everybody panics at the beginning of lab and runs in there and starts printing, that creates a huge hassle for everyone who's trying to print, as well as the poor tech guy in there who's always trying to keep the printers online. All right, Monday, the April 8th is a very important day. It is a complete total solar eclipse. There will be no class. We will be doing a test on that day. And remember how I said all classes, all tests will be taken in person in class, except for this one. Just this one exam. The rest of them, we will be in the classroom. All right, I am required to give you 10 minutes per hour of class time break. So we are section 14. Everybody open up Socrative, please. Which break schedule would you prefer? Oh, hush. Wow, it's neck and neck. The numbers keep changing. All right, final results, 40% say A and 60% say B. So we will have our break at 1:15. Let me set an alarm on my clock right now. Honey, I can't play with you right now, I'm sorry. 1:15 p.m. and that is going to be on Wednesday, break. All right, so in 34 minutes, we will have our break. What does that mean? Is somebody texted a question maybe? Chat. Oh, yeah, the cats. They get into everything, man. Okay, so blah, 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 blah. All right, first, some lab equipment. I will be giving you, this is the very first short quiz that I will be giving you in class. I am giving you, how do I get rid of this little bar at the bottom of my screen? Well, I don't know. I have a technology curse, so technology just like doesn't work for me. But anyway, this is the first of those short pre-lab quizzes that I am going to give you. And we will take this one on Monday, a week from today. Right? Quiz before class next time. No, next time is chapter 2, yes. So we're going to be taking this on Wednesday. Spelling does count in college. I'm sorry, it just does. You're a grown-up now. This is a beaker. It is for holding, mixing, and measuring. This is a flask. It is also for holding, mixing, and measuring, but instead of having a wide mouth, it has a neck. A flask has a neck. And then we have a graduated cylinder. Graduated cylinders are for precisely measuring tiny volumes of liquid. So you see all these little graduations here are different volumes that you can measure. And then it has this ring that keeps it from rolling. So take a picture of this slide with your phone to use to study with. Stare at it in your free time between now and Wednesday.
I don't mean to interrupt, but I can't see anything.
Uh-oh.
Am I the only one?
Is anyone else having that problem?
No.
I guess it's just me.
Can you log out and log back in?
Let's see.
All right. Also this, test tubes. These are for holding and mixing small volumes of liquid. We have a test tube rack for holding the test tubes. And then these little guys, these droppers, are called pipettes. And those are used for transferring liquids from one thing to another thing. So take a picture of this slide as well. And if you are not able to get pictures because you cannot see them, let's see. Oh, I can see the chat on my phone. How fun. That's very convenient. Okay. So if you can't see, type in the chat, drop your name in the chat, and oh wait, no, because I put this on. Yeah, if you can't see, drop your name in the chat and I will send you some pictures so that you can study. All right, let's talk about biology, the scientific study of life. How long do we have till break? It's coming up pretty quick here, isn't it? Oh, 31 minutes. Yes. Okay. So I have really shortened this lecture, this first lecture. I like to go into a little bit of detail about what life is. We're not going to do that, but I'm still going to tell you what life is, but we're not going to have as much of a discussion. Biology obviously is the study of life. So what is life? A living thing is anything that has all of these 7 properties. It has order, meaning that it is ordered on a cellular level. It can reproduce. And this one, again, this one is kind of iffy because if you get your dog fixed, your dog can no longer reproduce, but it is still alive. So reproduction is necessary for the species as a whole, not necessarily for each individual. But when I was teaching 8th grade, I gave that analogy, and this little boy in the front row folded his arms very seriously. He said, "Miss Heckman, when you fix a dog, he dies inside." Yeah, some knowledge there among the 8th graders. Growth and development, all living things are able to grow and develop. They can process energy. They are able to regulate a stable internal environment. That means that they can keep at a relatively constant temperature, blood glucose levels, blood sodium level, things like that. They are able to respond to stimuli. If you poke it, it will respond. And they are able to evolve and change through time. And the cell is the structural and functional unit of life. All living things are made of 1 or more cells. So some things meet some of these characteristics and others do not. So let's think about this. A drill, a battery-powered drill. It has order, but it's not cellular order. It can't reproduce, grow, or develop. It can process energy because it uses the energy from the battery, but it can't regulate. It does respond to stimuli. If you pull the trigger, it'll do its rotating thing, but it doesn't evolve or change through time. So all of these 7 characteristics must be met in order for something to be living. And now let's talk about the process of science. It is a method for studying the natural world using observations and testing, but you can't conclusively prove anything. This is a picture of Bigfoot. And, well, look at this evidence. Here we've got a picture of Bigfoot. So Bigfoot exists, right? Well, not necessarily. Just because you have a piece of evidence doesn't mean that the evidence is valid. And is there anything else on that slide? No, there's not. The only way to prove whether Bigfoot exists or does not exist is to simultaneously observe every area on the globe, because if you're just observing one area and you don't see him and then you move to another area, well, he could have been in a different area and then move to the area that you just observed, but you would think he's not there. So you have to observe everything all at once. And even then, we're assuming that he doesn't have the ability to become invisible. No living things that we know of can do that. But I mean, again, we're talking about Bigfoot here, so invisibility seems like it could be on the table. And a lot of people just use the words prove and they just kind of throw that around like, oh, yay, I proved my theory. You know, well, I mean, yeah, it can be used to mean that. But I do want you guys to be aware that there's always going to be a tiny element of doubt. Science cannot be used to test and explain everything. For example, we can only use science to test things that can be measured and observed. Here are two statements. The Killers are the best band ever, and the music of the Killers is listened to by many people. Which one of these is a scientific statement? It's going to be the second one, because the first is an opinion. You cannot measure or test an opinion, but you can measure or test a fact. The music of the Killers is listened to by many people is a fact, so we can test that using science. The existence of God is something else that science cannot be used to test or explain. That does not mean that God does not exist. It just means that if there is a God, he, she, they exists outside the realm of science, simply because God cannot be measured. You can't take like 1 cup of God and put it on a scale or something like that. So belief in the supernatural is compatible with belief in science. Now let's go over to Socrative, and we're going to do our next clicker question. Which statement cannot be investigated using science? Remember, our room name is NWAC Bio. And if we were in the classroom, I would have that written up on the board. Excellent. 92% chose C, which is the correct answer. My mother-in-law is crazy. This is not a scientific question because it does not contain anything that can be measured. How do you measure crazy? Crazy is an opinion. I think that person is crazy. I think that person is crazy. But what's crazy to one person might not be crazy to another. All the rest of these have measurable, quantifiable words. Unhealthy, we can define that. Heavier than average, we can find an average size, that's a number. Busiest, that means how many people are there, that's a number that we can find. But there is something that we could do to change this statement to make it able to be investigated by science. Does anybody have an idea how we could change number C to make it investigable by science? Okay, well, hopefully at least one of you is thinking that we could give a definition to the term crazy. We could take the DSM-4 or 5 or whatever the psychiatrists are on now, their little manual, and turn to a specific disorder, perhaps bipolar disorder. There's a list of criteria that you have to meet to be diagnosed with bipolar disorder. So we could change this statement to say, my mother-in-law has bipolar disorder. We can then open up that book and see if, she demonstrates the qualities on that list. Science, understandings of things and conclusions may change as additional data is gathered. For example, during the 17th century, people believed in something called spontaneous generation, which means that life arises from non-life. This is an actual recipe for mice from the 17th century. It says, if a soiled shirt is placed in the opening of a vessel containing grains of wheat, the reaction of the leaven in the shirt with the fumes from the wheat will, after approximately 21 days, transform the wheat into mice. Now, we know that this is ridiculous, but if you thought that non-living things could become living things, and you did this, and you saw the results, that there was no more wheat and the mice are present, it is a logical conclusion to think, well, the wheat must have turned into the mice. But because science is constantly changing our understandings of things, now we have what's called biogenesis, which means that life only arises from life. In the 1800s, scientists thought that protein was the hereditary material, which means the material transmitting traits from one generation to the next. In fact, it is actually DNA. And because we have this additional data and we've found that DNA is the hereditary material, this is a picture of the experiment they did. Because of that, we can build on that knowledge as well, and we can do genetic engineering. Scientific knowledge must have peer review and verification before being accepted as valid. Peer review means review by your peers, and verification is conclusion that the results are valid. This is very important because if every scientist just went around spouting everything that they thought, every hypothesis, well, that would be a huge mess. Here's a great example of that. This is a molecule called polywater. It was discovered in the 1960s as a form of water that was heavier and denser than normal water, and most notably could not support life. And it also had the ability to change any other water that it came in contact with into polywater. So this became a huge deal. Scientists were not able to replicate this one person's experiment. They could not get his results, and they kept trying and trying. Finally, one other person did it, but then he realized, this isn't anything different from just normal water. It's dirty water. So the scientist who had originally isolated this polywater had been sweating into his test tubes during his experiment, and he had isolated himself some dirty sweat water, which of course cannot sustain life. And so that's why peer review and verification are important. And now, that question is settled. The scientific community is okay with the non-existence of polywater. With new data and results, we can build on previous knowledge. We've already gone over this a little bit. We know that DNA is the hereditary material. We know what genes are. We know that genes can be manipulated, and that allows us to do some genetic engineering, which has some really beneficial applications for society, as we're going to see when we get to chapter 12. Scientific knowledge progresses in certainty, levels of certainty, from hypothesis to theory to law. Hypothesis is an educated guess. It does not have to be an if-then statement. Later on today, we're going to learn about two different kinds of hypotheses, a null hypothesis and alternative hypothesis, and neither one of them is an if-then statement. A theory is a well-tested explanation, multiple hypotheses that are supported coming together, forming a more complex explanation of a larger phenomenon. It must be supported by empirical data, which means numerical data collected by actual observation, and it has to be peer-reviewed before being accepted as a theory. Theories can also be modified and expanded. And then the final level is law. Law is an observation that happens every time under a certain set of circumstances, like the law of gravity, the law of opposites attract, that kind of thing. These days, we don't make laws anymore because we understand that the field of science is changing so quickly, and that the better technology we have, the more we come up with. And the more things we can see, the better microscopes we have, the smaller we can see. And we're always discovering new things. So our understandings are constantly changing. So it makes sense not to make a law because we understand that there's things we don't understand. So we only make theories these days. Scientific investigations have one of two purposes: pure science, which is science for the sake of knowing, and applied science, which is applying the pure science towards a practical goal. And within the scientific community, applied science gets a lot more funding from grants and from research institutions because obviously we're using it to solve real-world problems. But the issue is that applied science cannot exist without pure science also existing. A really good example of this is a spy probe was found crashed on some US land somewhere. And in that spy probe were some grains of sand. And because somebody had done a pure science study analyzing the grains of sand on many different beaches, the applied scientists were able, I'm sorry, baby, I can't. The applied scientists were able to go back to that pure science knowledge and apply that, and they could figure out where the probe came from because they knew the composition of the sand on these various beaches. I am not going to spend a lot of time on the scientific method. I found this on Reddit. I thought it was funny. I divide it into 7 steps. I'm sure you've learned it in many different steps, but it's sort of like a pizza. It's a process. It's a whole thing. It's one pizza, no matter how you slice it. You can slice it into like 20 pieces if you want, or 3 pieces. It's still a pizza. But I like to slice mine into 7 pieces. Observation, ask a question, form a hypothesis, do the experiment, analyze your data, draw conclusion, and communicate results. Communicating results is the most important step of the scientific method. And this is because if you don't tell other people what you know, how can they use your knowledge and your discovery to build on that for the betterment of science? Yes, it's a process that may be divided into a different number of steps. All right, and now we come to our 2 types of hypotheses. Experiments will always start with a question. Does X have an effect on Y? The first kind of hypothesis is called H sub 0, or the null hypothesis, which states that X does not have an effect on Y. And H sub A, which is the alternative hypothesis, states that X does have an effect on Y. And this information is all available to you. I have uploaded this entire PowerPoint into our Chapter 1 module. And so you can use that to look back on when you are doing the lab. Here's an example. Will students score higher on a test if they study 5 hours instead of 1 hour? The null hypothesis states that study time does not affect test score. X does not affect Y. And the alternative hypothesis says that it does affect the test score. So what results would cause me to reject the null hypothesis? Well, if you're rejecting something, that means you're seeing the opposite. So if you do this experiment and you see that study time does affect test score, then we're rejecting the null. To reject the alternative means we're seeing the opposite of the alternative. So if we do this experiment and we see that study time does not affect test score, then we reject the alternative. Here is another example. Does the type, oh, okay, well, cool. I was going to type in it, but I guess I don't have to. Does the amount of food my dog eats affect his weight? Null hypothesis says that it does not. And again, we always have to write a full sentence. The amount of food my dog eats does not affect his weight. Alternative hypothesis, it does. The amount of food my dog eats does affect his weight. I want you to notice we are not talking about gaining weight or losing weight. We are talking only specifically about affecting the weight, which means it could be a gain or a loss. When you are building your hypotheses, you have to speak to exactly what was written in the prompt. So you can't say, you could say change his weight because change and affect are synonyms, but you couldn't say it does not increase his weight or does increase his weight. What results would cause me to reject the null hypothesis? So if I see the null, if I see the opposite of the null, then I reject it. So if I do this experiment and I see the amount of food my dog eats does affect his weight, then I reject the null. And similarly, to reject the alternative hypothesis, if the amount of food my dog eats does not affect his weight, then I reject the alternative hypothesis. All right, let's go back to Socrative. Remember, our room name is NWAC Bio. How many people, I guess I'm going into my classroom mode. How many people, show me your hands, have seen Scott Pilgrim versus the World? If you have not seen this show, you should watch it. It's very, very cute. Little baby Michael Cera and his cute little blue-haired girlfriend are sitting in the basement eating garlic bread. And Michael Cera says, I could eat garlic bread my whole life. And Ramona says, well, bread makes you fat. And of course, you know, with his mouth stuffed full of bread, he says, bread makes you fat. So we're going to see what the null hypothesis would be for this experiment. If the experiment is designed to test if eating more bread makes you gain weight. So go over to Socrative and type in what you think would be the null hypothesis for this experiment. Remember, the room name is NWAC Bio. I'm going to write that on here since I can't write it on the board. Okay, we've got some good answers here. You do not have to get creative with your wording. Hypothesis is just a simple statement. Again, it's written as a statement. It's not a question, it's a statement. There's no difference between if you eat the bread or use it for an estimate. No, that's not the null hypothesis, because null hypothesis, I see a lot of alternative hypothesis here as well. The null hypothesis states that eating bread will not make you gain weight. I think we're at a disadvantage here because we're not in the classroom. Eating more bread does not make you fat. Bread does not make you gain weight. Yes, those are the null hypothesis. Somebody wrote, can consuming different variations of bread will affect more weight gain? That is a good experiment, but it is not the null hypothesis for this experiment. Eating more bread could not make you gain weight. Very good. All right, so let's go ahead and type that in. The null hypothesis, eating more bread does not weight gain. Now let's look at the rejection of the null hypothesis. If the null hypothesis states that eating more bread does not cause weight gain, let's do another short answer. Here we are. Then what would cause us to reject this? Yes. Yes. So if you do this experiment and you see that in fact, eating bread does make you gain weight, well, then you are seeing the opposite of the null hypothesis, so you have to reject the null hypothesis. Weight gain. Bread does cause weight gain. Gaining weight from eating bread. Very good. All right, now let's do another one. The alternative hypothesis. Ooh, it's almost break time. What would the alternative hypothesis be? Good, we've got some really good answers here. If eating more bread does cause weight gain, that is the alternative hypothesis. So not consuming bread will not make you gain weight. That is not the alternative hypothesis because our hypotheses don't start with the word not. They're just statements about what is. Not gaining weight, not consuming bread, eating bread would make you gain weight. Yes. So the alternative hypothesis is that eating bread does cause weight gain or does make you gain weight. So there's our alternative hypothesis. Now, finally, if that is the alternative, then what would we need to see if we actually do this experiment? What would we need to see if we're going to reject the alternative? Excellent, yes. If eating bread does not cause weight gain. So the alternative hypothesis remember, stated that eating bread does cause weight gain. So if we do this experiment and we see that eating bread does not cause weight gain, then we are rejecting the alternative. Yes, very good. Eating bread does not make you gain weight, does not cause weight gain. People gaining weight is a result of eating more bread. There we go. Eat healthy bread. Good idea. Okay. Oh, all right. We are on to the next section. Let's see how long this is. Wow, we're almost done. Excellent. Very good, very good. Okay, and then we can move into the lab. All right, so why don't we, I don't want to start. Nah, we're going to start. Let's make use of our time. All right, designing a controlled experiment. There are 3 types of experiments that we're going to discuss in this class. An observational experiment where nobody changes anything. A simple comparison experiment where there's no control, you're just comparing individuals to each other. And a controlled experiment where you're comparing all individuals to one standard. And within this class, we're mostly going to do controlled experiments. But let me tell you about these a little bit. An observational experiment is where you just set an event in motion and watch what happens. This is really good for studying things like endangered species, where you can't be very hands-on. Simple comparison, controlled experiment. Yeah, we'll go over those. There's 4 parts to a controlled experiment. First, you have the independent variable, then you've got your dependent variable, you've got your control, also called the control group, and the confounding variables, which are variables held constant. Sometimes these are called constants. And before, before we dive into that, it's only 3 minutes till break. So let's go ahead and break, reconvene at 1:30. And we will finish up and start the lab. Prozac, what was that? 12:51. What was happening at 12:51? All right, it's 1:30. Let's go ahead and get started again. I saw in the chat, Danielle, you put your name. Is that because you didn't get pictures of the things that there will be the quiz on next time? Are you back yet? Okay. Can people hear me? Hello?
Yes, I can hear
Yes.
you.
Okay, cool.
Yes.
All right, so maybe she's not back yet then. All right, cool. My computer's working somewhat. All right, well, I was gonna bring those back up, but now I will not. So let's go back down to experimental design. Woohoo! Controlled experiments. The independent variable, we also call the manipulated variable, and this is the cause of whatever you're changing. So right now we're talking about parts of a controlled experiment. Remember, we had the 3 different kinds of experiments. The independent variable is the cause. The dependent variable is the effect. That's what you measure to see if anything happened. And to help you find them, you can insert into this sentence. Does independent variable cause a change in dependent variable? You could also use this way longer sentence, but I don't like that one as much. So example, trees receiving the most water produce the largest lemons. Let's identify the variables here. What are the variables? What is changing? The amount of water and the size of the lemons. So which one of those is the cause? The cause is going to be the independent variable. So that would be the amount of water, and the effect is the size of the lemons. So that dependent variable is the size of the lemons. Let's do another one. We also have a control is another portion of a controlled experiment. This is a group without the independent variable. Let's see, somebody's got their mic on. Can you please mute yourself? Okay. Oh, Danielle, okay, you're back. Cool. So you put your name in the chat, Danielle, because you needed the answers to the quiz for tomorrow, but not tomorrow, Wednesday, I assume. Okay, cool. All right, we'll get your camera ready. We'll go back up in just a second when we finish this slide. We'll go back up and I'll show you the answers. All right, control is a group without the independent variable. It is something that you have not changed that you're going to compare your results to. And then the final component is the confounding variables. Sometimes we call these constants. These are things you're not testing that could get in the way of the results if they were changed. You don't want them to get in the way of your experiment and affect or confound the results. So we're going to go back up to those quiz answers for anyone who didn't get to see them, and then we will do an example. Here is the first slide. Take a picture. All right, are we ready for the next one? Here's the second slide. Take a quick picture of that. All right. And back down to our example. More pounds of potatoes will be produced if the soil is fertilized more. So this is similar to a question that I would give you on an exam. I would give you a scenario and ask you to identify the portions of the experiment. So let's try that right now. First, we identify the things that are changing. That's the variables. The pounds of potatoes, that's one variable. And the amount of fertilizer is another variable. Let's see which one is independent. Which one is the cause here? The cause is going to be the amount of fertilizer in the soil. When you are talking about your variables, you have to be very specific. You cannot say soil because that's not specific enough. If you say your variable is soil. Well, what about the soil? The color, the composition? Again, you can't just say fertilizer. What about the fertilizer? Are you testing the nitrogen content? Are you testing the brand? You've got to be very specific. We are using the amount of fertilizer in the soil. That makes the other variable the dependent variable. So that would be pounds of potatoes. And again, you have to be very specific. You cannot say potatoes. That is an incorrect answer. And pounds is also an incorrect answer. Because if you just say potatoes, well, what about the potatoes are you measuring? Are you measuring their length? Are you measuring how many eyes they have? Are you measuring, how big around they are? And again, pounds of what? Pounds of fertilizer, pounds of soil? No, pounds of potatoes. The control is going to be a group that does not get the independent variable. Oh, yeah, also we could use this sentence to insert their variables to see if which one is independent and which one is dependent. So let's go ahead and use that. Does more pounds of potatoes cause a change in if the soil is fertilized more? That makes no sense. Let's try again. Does, if the soil is fertilized more, cause a change in more pounds of potatoes? That makes sense. So now we know that the independent variable is the amount of fertilizer and the dependent variable is the pounds of potatoes. Control is going to be a group that does not receive the independent variable. So that's the soil without fertilizer. That would be potatoes grown in soil without fertilizer. So you're growing them in soil without fertilizer, that's their control, and then soil with fertilizer, that's the experiment. And then you're going to compare them. Confounding variables, things you are not testing that could change the outcome if they were changed. Things you'll have to keep the same. Well, all of your potatoes are going to have to get the same amount of fertilizer. If you give some of them different amounts of fertilizer, then at the end, you don't know if your measurements are going to be because you gave them different amounts of fertilizer or if you, oh, wait a minute. Oh, shit. Sorry. That is not for this one. No. The amount of fertilizer is the independent variable. So we can't use that as our constant. What's another good one? Okay, sunlight. All of the potatoes will have to receive the same amount of sunlight because if you put half of them in the shade and half of them in the sun, well, the ones in the shade are probably not going to grow as well. But you're not testing shade. You're testing the amount of fertilizer. So we could say sunlight, species of potato. Yeah, we would have to use the same species of potato for all of them. Give them all the same amount of water. These are all confounding variables or constants. Null hypothesis states that more pounds of potatoes will not be produced if the soil is fertilized more, meaning the amount of fertilizer will not change the pounds of potatoes. And the alternative hypothesis is that the amount of fertilizer will change the pounds of potatoes. All right, you try this one. Go ahead and write it down on a piece of paper, or if you are somehow typing on the PowerPoint that I had put up in the Canvas shell, you can use that. I'll give you guys, I'm set the timer for 5 minutes. And then we'll talk about it when it goes off. We got 1 minute left. All right, let's talk about this. The growth rate of a pine tree depends on how much carbon dioxide it receives. The amount of carbon dioxide and the growth rate of the pine tree, those are the two variables. Those are the things that are changing. Which one is the cause? That's going to be the amount of carbon dioxide. The effect that what we're measuring is the growth rate of the pine tree, growth rate of the tree. So that's the dependent variable. Our control is going to be, well, in this experiment, it could be one of two things. It could be a tree with unchanged atmospheric carbon dioxide levels, or it could be a tree with 0 carbon dioxide. You could surround it with an environment that completely prevents any carbon dioxide from getting to it. It would die, so that's probably not a good control, but you could do that. For the purposes of this experiment, I would accept that as an answer. And they actually do this experiment up north where there's a lot of pine trees. People take massive amounts of carbon dioxide, just dump it on plots of pine trees and see what happens. And surprise, surprise, they actually do not grow slower. They grow faster. They do grow faster. Let's see. Some confounding variables. They will all have to be the same species of tree. You will need all of your trees to get the same amount of sunlight and the same amount of water. Null hypothesis states that X will not have an effect on Y, so the amount of carbon dioxide a pine tree will not change its growth rate. And then the alternative hypothesis states that the amount of carbon dioxide a pine tree receives will change its growth rate. All right, are there questions about this? Okay, so that brings us to the end of the lecture portion. Now we're going to start. I always like to incorporate some worksheets into my lectures because I think that interactive worksheets, especially this, like obviously we can't be interactive today, but group work is very important in helping people understand things. So let's go down here to modules. We'll go over to modules in our Canvas shell. And let me go to student view. There's our lab quiz if you need to take that. Chapter 1, okay, variables and experimental design practice. So this is something that I put together to help you practice the identifying the variables and the different parts of the experiment. And I'm going to give you about 20 minutes to work on this. And then we're going to go over it as a group. And after that, we'll talk about the chapter 1 assessment question. I guess I should download that as well, huh? So we can go over it. So I've got 20 minutes on the timer. Just get as far as you can. If something stumps you, skip it and move on. Oh, you can't write on that. Yes. And I'll be right here if you have questions. Is it safe to assume that there's more than one independent variable and or dependent variable, like questions 2 and 3? Which question are you looking at, Mason? In each of these questions, there is one correct answer for independent variable and one correct answer for dependent variable. Okay, corn seeds, yeah. No, there's only going to be one independent variable for each question and one dependent variable for each question. You do not need to edit the document and submit it, Sarah. We're going to talk about it as a group in a little bit here in 7 minutes and 53 seconds. 2 minutes. All right, let's talk about this. Enable editing. Okay, this file is untrusted, whatever. All right, identify the independent variable and dependent variable. Number 1, apple trees grown in full sunlight will produce the largest apples. First, we identify what's changing. That would be the amount of sunlight and the size of the apples. So here, the cause is the amount of sunlight. Again, you can't just say sun. What about sun? You can't just say amount. Amount of what? And you can't say full sun because the amount of sun is what we're measuring. And dependent variable is going to be the effect, the apple size. You can't just say apples. What about the apples? Number 2, and if you have questions, of course, just pipe up at any point. If we were doing this in a classroom, it would be easier for me because I would be able to see confusion on your faces and solicit questions, but I can't do that right now. So you'll have to come forward with your own questions, which I know can be intimidating, but it's a learning process. All right, number 2, an investigation was done to see if corn seeds would sprout at different times depending on the temperature of the water in which they were placed. All right, so what's the cause of the change here? The cause is going to be the temperature of the water. And the effect is going to be time to sprout. Number 3, the amount of pollution produced by cars was measured for cars using gasoline containing different amount of lead. What's the cause of the change here? The cause is going to be the amount of lead in the gasoline. You can't just say gas. What about the gas? You can't say lead. What about the lead? Amount of lead in gas. And the dependent variable, what's the effect that that is having? That effect is the amount of pollution produced. All right, questions about those 3? Okay, let's Let's move on to number 4. 3 sunflower plants are watered with salt water. Gosh, I feel bad for those plants. Each plant receives a different concentration of salt solution. A 4th plant receives pure water. After a 2-week period, the height is measured. What is the independent variable? The independent variable here is going to be the cause of the change. So that would be the amount of salt in the water. You could also say concentration of salt in solution. What is being changed? What are we measuring? The dependent variable that we are measuring is the height. The height of what? Of the plant. A causable control. So that is, remember from the slides, a control is a group without the independent variable. So this would be a group without salt in the water. So flower grown without salt in the water or with pure water. And then that's what we're going to use to compare our results to. And a possible constant, some things we need to keep the same. Same species of flower. They all need to get the same amount of sunlight. The same volume of liquid. They all need the same fertilizer if we're using that. Questions about number 4? Let's see if there's anything in the chat. Nope. Okay. Number 5. 1 tank of goldfish is fed the recommended amount of food per day, according to the instructions on the fish food package. A 2nd tank gets twice the recommended amount of food, and a 3rd tank gets 4 times the recommended amount. For 6 weeks, the weight of the fish is recorded daily. What is the independent variable? The independent variable, the cause, is going to be the amount of food given to the fish. The dependent variable, the effect, is going to be the weight of the fish. Control, which would be a group without the independent variable. So if you're actually doing this experiment, the control is going to be the tank of goldfish fed the recommended amount of food per day. You could also, for the purposes of the exercise, I would accept the answer fish not given food. That would not work in a real life scenario, but for the purpose of the exercise, I would accept that. Some constants. What are some things we need to keep the same? The species of fish. We need to keep the brand of food the same. Be them all the same brand. They all need to be in the same kind of tank, etc. A possible null hypothesis. Possible null hypothesis would be that amount of food does not have an effect on weight of fish. So notice how I've phrased this. Amount of food, that is the independent variable. Weight on fish, weight of fish, that's the dependent variable. So essentially what I'm saying here in my null hypothesis, I'm always going to say independent variable does not have an effect on the dependent variable. And the alternative hypothesis is that independent variable does have an effect on dependent variable. So now we can just go in and replace these independent variables. The amount of food does not have effect on weight of fish. There you go. Alternative hypothesis. That would be independent variable does affect dependent variable, which would be amount of food does affect weight of fish. If we reject the null hypothesis, that means we're seeing the opposite of the null hypothesis. So here's our null hypothesis. If we do this experiment and we see that the amount of food does have an effect on the weight of the fish, well then, we reject our null hypothesis. To accept the null hypothesis then would be if we see the same as that hypothesis. So if we do this experiment and we see that the amount of food does not have an effect on the weight of the fish, then that will allow us to accept the null hypothesis. Questions? Okay. So that brings us to the end of the lecture. I have a final assessment question. Where's the PowerPoint? There we go. Right here. And if you go to Canvas, you can find it. Let's go back to modules. Chapter 1, experimental design. There it goes. Chapter 1, assessment question. Until 3:30. Why did I lock it until 3:30? Well, I'm gonna have to unlock it then. Oh, okay, yeah. I'll unlock this. Let's do that. What time is it? I'll just say from 1:30 to p.m. All right, so this is due at the end of class. Oh, switching the null and alternative. No, that is not okay. I'm glad that you noticed that. That's very important. So the null, you think null is you can think of it as zero. Zero effect, no effect, null effect. And alternative is the opposite. Yes effect. All right, so go ahead and do the chapter 1 assessment question. And then the lab. Actually, why don't you do the chapter 1 assessment question? I'll make that due at midnight so we can move on to the lab right now. Okay. So let's move on to our lab, shall we? Now, if you have the lab handout, that's great. If you don't, do not fret, because over here in modules, we're going back to chapter 1. There's the PowerPoint there. There's our assessment question, lab 1, scientific methodology, online/remote. If you have been unable to print, you may type your answers into this document and you can download right there. So that's a typable version of the lab handout for lab 1. You will have to hand draw a graph and upload a picture of that. What you're going to do for the lab is you're going to read all this, follow these instructions. There's some videos you need to watch. And then at the end, you're going to turn in pictures of your handout. Let's go over real briefly. I have put this is what we're going to do right now, the PowerPoint presentation. I've put that up here for you to have for reference. We're going to do that right now. On page 4, you can skip page 4 today. You don't have to do that. On page 5, you're doing a preliminary observational experiment, which is what the videos are. And then next, you're going to design a controlled experiment to test your hypothesis. Remember, we've got the independent variable, which is the cause. The dependent variable, that's what's measured. That's the response, the effect. Control, the natural state, or we could say group without independent variable. And constants are things you're not testing that might affect the experiment. God, I need water. Excuse me. You're going to make two hypotheses, a null and an alternative. Null states no effect. Independent variable does not affect dependent variable. Alternative states it predicts yes, an effect. Independent variable will affect dependent variable. So this is what would be on the lab cart if we were in lab. We would have our graduated cylinders for measuring water, thermometers for measuring temperature, and a beaker for measuring and holding water. And you would get all of these things. You would get 2 beakers. You would get 2 Alka-Seltzer tablets. Each packet contains 2 of them. And you would measure out some water into the beaker and then take its temperature and then drop the Alka-Seltzer tablet into it and measure the time it takes for the top of the water to clear. You do not want to wait until all the bubbles are gone because you will be waiting 2 hours. Just wait until the top of the water clears. And that will be your time. And then on the next page, you're not going to graph your data. This is some sample data that they gave you. This is not the data from the videos. This is completely different. And this is what you're using for the analysis. So you're going to compute an average time for these guys. And then you're going to create a graph based on that data. When you are making your graph, I will be grading you on 4 things. All graphs have 4 things. The first is a title. It might be long. It goes along the top of the graph. So in this case, the title might be something like the effect of temperature on dissolved rate of Alka-Seltzer. That's long, but it accomplishes the purpose. It tells you what is in the graph. The point of the title is that if you read the title, you can know exactly what the graph is showing you without having to read the rest of the lab report. Then you have labels on your X and Y axes. The independent variable always goes on the X-axis, and the dependent variable always goes on the Y-axis. Third, we always have our units. They are in parentheses next to the axis labels like this. And fourth, we have our data. If you are using a line graph, you must connect the dots. You will be graded on that. So these are the 4 things that all graphs have. Make sure that your graph has them as well. On page 7, there is a typo. It says, why is it important to collect more than one subdata for each temperature? Why compute an average temperature? What they mean there is time, because it even says it up here, average time. You're computing an average time, not an average temperature. And then finally, upload a scan of pictures of pages 5, 6, and 7 to Canvas. These are the only pages that I need. And that's the cleanup, but we don't need to do the cleanup, obviously. So I will be online for the rest of until 2:45. And this is all due by midnight. So if you have any questions, feel free to ask. See if there's anything in the chat. What happened to switch the null and alt? Oh, well, we already went through that, I think. Yeah, okay. All right, so good luck. I'm here if you have questions.